actually when you just try to organize immune cells based on their overall transcriptomes. If you're just using long read, all of a sudden we're finding entirely new cell states.
There is a difference between these different alleles.
Okay.
And when we sequence through with PacBio long read sequencing, so now it's pure target.
Sure.
So what we saw was the siblings that that were unaffected had a pure repeat expansion of this pentanucleotide repeat A, T, T, C, T.
One of the very most difficult aspects of lens crystallins is that there are about 18 to 25 of them. Some of them differ by 1 or 2 amino acids. The short read approach doesn't work because you're basically chopping them up and you can't reassemble them. So the only other way to do it, until we had the long read approach with the Vega, was to do it with the old school, clone the cDNAs, put them into plasmids, put them into vectors, do the screening, so on and so forth.
Hello everyone, and a warm welcome to the PacBio workshop at this year's PAG conference. My name is Jonas Korlach, and it is a great pleasure to once again be the host of today's event. You have just seen the trailer of a new video series that we're launching this month. There's a QR code that gives you more information, and the purpose of it is to highlight and amplify all the great work that you do, all the great science, but also to give a picture on the personalities and the people behind that great science. And this is also the theme for today's workshop. You will hear from 5 researchers applying PacBio HiFi sequencing in a diverse spectrum of different applications. Their presentations will be preceded by my colleague Jake Brandenburg telling you about the latest products and application and what's to come in 2026. Before I give the stage to the speakers, please allow me to make a few introductory remarks. As I look around the room, I see many familiar faces, and I know from our discussions and from the publications that many of you have now generated a high-quality, haplotype-resolved genome as the architectural blueprint for your favorite species. And both Jake and also Jeremy Schmatz will tell you how that can be done now even more efficiently and economically using our latest SparkNext chemistry. But of course, the blueprint is just the beginning of the scientific understanding of the true biology, and so it is now necessary to add a variety of multi-omic workflows and applications and data sets to truly understand the complexity and the beauty of your organism, adaptation, evolution, disease resistance, and all the things that you do. And so for that, there's HiFi for that. And there's HiFi for that is the theme of our presence at PAG 2026 at this conference, showcasing that there are now kits and products and workflows that are available for all these multi-omic data sets. So whether you're interested in chromosome architecture or epigenetics, both with regard to methylation as well as chromatin or the transcriptome, we now have workflows that give you an insight into the inner workings, as it were, of the organism. If you're not familiar with some of the abbreviations on the right, don't worry, you're in the right place, as Jake and all of our speakers will talk about this in a lot more detail. You see on the bottom left that I have highlighted isoform resolved transcriptomes, and I want to spend just a couple of minutes on this because I feel that this is not as appreciated in the scientific community as compared to, for example, the difference between a short-read genome assembly and a long-read genome assembly. And that is to say that the term gene annotation and gene expression is really far too low of a bar to understand the biology of an organism, and it can be misleading relative to what is needed here. And well, why is that? Well, in humans, where we understand this best, it is known that 95% of all multi-exon genes undergo alternative splicing. The average number of unique transcripts per protein coding gene is around 7. Moreover, these alternative spliced isoforms do not have just slightly different functions. In fact, their functions are so different that they behave as if they were encoded by distinct genes. And there are now many examples where two isoforms from the same gene have the opposite biological function. BCLX, the first one, is very well known. It's in all the textbooks. But full-length RNA sequencing, or Iso-Seq, has demonstrated many more examples where isoform 1 causes a certain function and isoform 2 causes the opposite function. So if you're just counting expression and if you're just looking at the gene, you really haven't learned anything yet about how that is affecting function. So of course it is gene isoform annotation and expression that is the relevant metric to understand the organism. And you cannot see this with short-read RNA-seq. This has been commented on by the community as early as 2013. Ian Korf, in a review, compared short-read RNA sequencing to going to a magazine stand, shredding all the magazines, and then trying to put that back together. Mike Snyder commented that short-read RNA sequencing is, quote, a really crazy way of doing things. And then more recently, Colette Felton, a postdoc at UCSC, compared short-read RNA sequencing to this famous fable of the blind Buddhist monks that touch an elephant and trying to reconstruct the elephant. And she said that with PacBio, you just see the whole elephant. So there are many examples now where Iso-Seq has demonstrated substantial improvements in genome annotation, perhaps starting with Richard Kuo and Dave Bird and Alan Archibald back in 2017, demonstrating that the chicken transcriptome is just as complex as the human transcriptome. So all the things I talked about before, they all hold for plants and animals, certainly. A series of papers by Guoji Zhang and also covers of Science and Nature with Evan Eichler looking at non-human primates and finding in some cases over 1,000 previously unknown gene models that were all supported by Iso-Seq transcripts. And then remarkably, even organisms that have been intensely studied genetically for 30 years, like the zebrafish. 6 months ago, there was a paper by a Chinese group finding over 2,000 previously unannotated genes and 33,000 new isoforms of previously annotated genes, thereby substantially expanding the current zebrafish gene annotation. And then more recently, about a month ago, a T2T assembly of the zebrafish and complete re-annotation of the zebrafish genome, again expanding the repertoire of alternatively spliced RNAs by 68%, and in some cases breaking the rules that NCBI and RefSeq have set in their databases. So there are over 250 genes in the zebrafish that have more than 50 isoforms per gene, and that was the arbitrary threshold that was set in RefGene. If you look at the paper, the record holder in the zebrafish is a brain-related gene that has 48 exons and 970 individual isoforms from just that one gene. So we have to rethink, and admittedly, it was too expensive and the throughput wasn't high enough in past years, but now with our Kinnex products and solutions, which Jake will talk about more, and Shenna as well, this has become very efficient and economical. So now returning to the analogy, beyond just looking at one species deeply, you also want to now scale to the population scale and get insights, and there's HiFi for that too. So you'll hear from our presenters, whether it be pangenomes, low-pass resequencing of many individuals, or looking at symbionts and the microbiome. We in pink have developed high-throughput automation solutions, and then in blue, working with our partners, for example, Sequel with the Longplex kit, or software like Panagram from Mike Schatz or Kufu from Veil Genomics, making the analysis straightforward and getting insights on the population scale. So we now have efficient, high-throughput workflows from extraction to analysis to support your research. And then there are insights possible with the availability of large numbers of species that, frankly, I find just amazing and have been unimaginable just a few years ago. This is a very recent preprint from about 2 weeks ago by the Sanger Institute and the Darwin Tree of Life Consortium, leveraging 708 samples from 661 species, HiFi assemblies from Britain and Ireland, and looking for germline and especially somatic mutation processes. And they find that there's a far considerably greater diversity of mutational processes across the tree of life than found in human samples. So in the paper, they identify something like 95 mutational signatures. 2 of these had previously been described in human cancers. And remarkably, they cluster with regard to the classification, so they're clade specific, telling us something really new about adaptation and so forth. And I really like the last sentence of this paper that reads, if this represents the diversity present in species resident on a few temperate rocky islands in the northwestern fringes of Europe, we can only wonder at what might await discovery planet-wide. So in that spirit, let's have a workshop. We're going to have great presentations, and we're going to see lots more discoveries. And with that, we'll start the program, and I'll ask my colleague Jake to come to the podium to tell you about the latest products and applications.